Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, in this chapter, Cell the Unit of Life, in the previous class, we were discussing about the nucleus in detail, and we have stopped at this slide in the previous class. From that, we are continuing today in this class. And in the previous class, we have already talked about the different type of chromosome, that is metacentric, submetacentric, acrocentric, and telocentric. Here, in this chromosome, these are the some special type of chromosome. In some chromosome, you will see that non-staining secondary construction. See, primary construction is secondary, primary construction is centromere. Secondary construction is found at some constant region, and that give a appearance of a small fragment called satellite. Okay, so this is the satellite, but secondary construction is the also known as nuclear organizer region, and this one. This is primary construction. Okay, so secondary construction or nuclear organizer region has a genetic DNA which has the ability to form rRNA, and this rRNA help in organization of a nucleolus. So because of this, we call it as a nuclear organizer region. Now, after this, we are going to talk about the how the chromosome are formed or condensed. Chromatin get condensed to form a chromosome, and this for this the histone protein are required. Now, let's see how they work. So, typical nucleosome contains. So, nucleosome is formed by the DNA when coiled around the histone protein. So, nucleosome contains 200 base pair of DNA double helix. The double helical 200 base pair DNA is that which wrap and take two turns around the core of histone, which wrap and form a two turns around the core of histone octamer. And this histone octamer have two copies of each four type of histone protein, that is H2A, H2B, H3, and H4. So here histone protein has H2A, H2B, H3, H4, and they are both in a subunit dimer. So this dimer will look like this. So octamer is there now. Each has a dimer. So H2A, H2A, H2B, H2B, H3, H3, H4, H4 octamer is there. And over octamer, the DNA double helix is coil. So DNA will take two turn, actually 1 and 1/4 turn. DNA takes, if you talk of DNA takes 1 3/4 turn around the histone octamer. And this DNA is of 200 base pair, which coil around the histone octamer. Now, histone octamer and DNA coil around the one histone octamer, and when it coil around the another histone octamer, these two histone octamer are connected by another one histone protein, that is H1 histone protein, and this molecule lies outside the nucleosome core, means it is outside the histone octamer, and it seals the two turns of DNA by binding at the protein where DNA enters and leaves the core. Means where the DNA enters over the histone octamer, and when it leaves the histone octamer, at this point the DNA H1 protein will bind, and this H1 protein helps to seal the two turns of DNA. Now, the DNA that continues between the two successive nucleosomes is called linker DNA. So, this H1 protein histone is linked with this linker DNA. And linker DNA is nothing but a DNA between the two successive nucleosomes. So, the association between the negatively charged DNA and positively charged histone allows the meaningful DNA packaging inside the nucleus. Inside the nucleus, the packaging of this DNA takes place. And you will see that this will look like this. See, this is H2A, H2B, H3, H4, and this is a dimer, and over which the DNA takes 1, 2 turns or 1, 3, 4 turn, and this is the H1 protein which seals the entry point and exit point. Okay, and this DNA is known as, just connect the two nucleosomes, it is known as linker DNA. Now, the beads on a string. Now, when the several nucleosomes are formed in the chromatin region, they will appear like what beads on a string structure in chromatin. Is packaged to form a chromatin fiber that are further coiled and condensed to form a chromosome. So, chromatin further coiled to form a chromosome. See, this is the one of the important proteins. If we take a DNA, then DNA condense and coil around histone and form one nucleosome. See, this is one nucleosome, this is another nucleosome, this is another nucleosome. Then when it coil further, then it will form a chromatin. When chromatin get condensed, it is going to form a chromosome. Okay, So such particular thing are seen in this. Hmm. Okay, so that is all about the what particular nucleus. Now we are going to talk about the number of chromosomes in this nucleus. So if we talk about number of chromosome, then this number of chromosome varies from species to species. Varies from species to species, but fixed for a particular species. Means if it is a Homo sapiens, we know that there is a 46 chromosome. Then in every individual of Homo sapiens, there is going to be 46 chromosome. So if we talk about a least chromosome number, in plants is 2n is equal to 4, so n is equal to 2 in Haplopeppus gracilis, and Haplopeppus gracilis is an example of composite family, whereas the highest chromosome number are 2n is equal to 1260 in ophioglossum, and ophioglossum is a pteridophyte, and this pteridophyte is called adder's tongue. Okay, in that the 2140 60 chromosome are found in the diploid cell. Now here n represent one set of chromosome or haploid or gametic chromosome number. So haploid has one number of chromosomes, whereas one set of chromosomes, whereas 2n represent diploid or somatic number. Now after this, in eukaryote, two type of chromosome are found. Eukaryotic chromosome, if we talk, this eukaryotic chromosome are of two type, depending on based on sex and body character. And this eukaryotic chromosome are of two type. That is autosomes and sex chromosome, or heterosome or idiosome. If we talk about autosome are They are also known as non-sex chromosome. They are non-sex chromosome. They are non-sex chromosome means they are not have a role in identification of sex of an individual. So they are also known as somatic chromosome, and they have nothing to do with the sex determination. So we can say no role in sex determination. Just they involved in understanding or determining the body characteristics. But if we talk about the sex chromosome or heterosome, they are they have a role in sex determination, and they are of two type, X and Y. So these are the two chromosomes. Now if we talk about the function of chromosome, function of chromosome, and if it is made up of a function of chromosome, it is known that chromosomes are responsible for carrying the genetic information because they have DNA. So they are responsible for genetic information from one generation to another, and genes are located on them. And genes are located on them. Okay, so these chromosomes are responsible for carrying the genetic information from one generation to the another generation, and the genes are located on them. Okay, so they are passing from one chromosome pass from one generation to another generation through the gametes and carry the genetic information. And any variation in chromosome will lead to change in the structure of organism, will lead to change in character of an organism. Okay, so this is all about the nucleus and the particular the what is chromosome and based on the centromere, the type of chromosome and all these chromosome we have discussed. Now we are going to talk about the special type of chromosome. Okay, and if we talk about special type of chromosome, then the special type of chromosome are also known as giant chromosome. Okay, they are the special type of chromosome or the giant chromosome. Now, why they are giant chromosome or special type of chromosome? Because they are large sized. So we can also call it as large sized chromosome. Okay. So in some organism, the chromosome assumes specific structure in specific tissue and they become large sized. Okay. And this giant chromosome are of two types. Number one, that is Lembrish chromosome, and number two, that is Polytony chromosome or salivary gland chromosome. Salivary gland chromosome or Polytony chromosome. Okay, these two type of chromosome are found. So salivary gland chromosome or Lembrish chromosome are found. So if we talk, we are going to discuss this two type of chromosome one by one. So first we are going to discuss the Lembrish chromosome. Okay, and if we talk about this Lembrish chromosome, these are described by Racket. Okay, and this Lembrish chromosome, if we talk first about its location, they are present in primary oocyte nuclei. Oocyte is the cell which is going to undergo oogenesis. So this primary oocyte nucleus of vertebrates as well as invertebrates. So they are large size and they are found in this primary oocyte. Now, this Lembrish chromosome are observed during diplotin stage of prophase 1 of meiosis 1 as bivalent chromosome. Bivalent means they are in a pair. Okay. So the question is why these Lembrish chromosome are large sized? Because they are observed in diplotin stage, and in diplotin stage chiasmata formation occur. So in diplotin stage chiasmata formation occurs. If we talk about this chiasmata, if we talk about this, when the homologous chromosome are in a pair, and when they this homologous chromosome after pairing, when they start separating, they form this X-shaped structure. So this X-shaped structure, it is known as chiasmata. And when during chiasmata, if we observe this X-shaped structure during chiasmata, when we observe this X-shaped structure, it look like this in Lembrish chromosome. They are look like this. So they look like this. So when we take one loop, these are known as lateral loop. And when we draw the structure of one lateral loop, that lateral loop will be look like this. So this lateral loop appear like this. So the point from where this lateral loop originates, it is known as chromomere. It is known as chromomere. And from the chromomere pair, you will see the origination of this DNA. So this DNA will appear like this loop. And if this loop is free, it is going to form a protein and RNA over this. So some protein and RNA are formed like this. So these are the proteins and these are the RNA. So here this proteins and RNA are there. This RNA is the actually messenger RNA, and this mRNA and protein combinedly known as informosome. And this informosome ha has a information which used during embryo development. Okay, so this mRNA and protein, which are known as informosome, which are used for development of embryo. Development of embryo. So this is the what particularly we can say about the informosome. Okay. So here the main axis, it is made up of DNA. DNA form RNA, and RNA has a proteins, and that all particularly form informosome. So such loops, lateral loops, or several lateral loops are there. So if several lateral loops are found in whole these two chromosomes, they are going to have obviously a larger size. And this larger size, due to this larger size, they are known as special type of chromosome, Lembrus chromosome. And obviously you can see they have a brush-like appearance as well. Because of this, they are known as Lembrus or test tube chromosome. Okay, so Lembrus chromosome, or we can call it a test tube chromosome. Whereas another type of chromosome which are found is the second type of chromosome, that is salivary gland chromosome, or polytenic chromosome. So salivary gland chromosome or polytenic chromosome are also a large size chromosome. Okay, so these are found in salivary gland cells of order Diphthera of class Insecta. And this special chromosome reported by E.G. Balbiani in 1881. Now, it is due to presence of this large chromosome, due to presence of this giant chromosome, maximum cytological studies maximum cytological studies have been made in Drosophila. Another example is Chironomus, in which the salivary gland chromosome has been observed. So, if we talk about the salivary gland chromosome, then this giant chromosome, if present, they are found to be attached with each other like this. Okay, and if they are attached with this like this, then and if we draw this segment, then this segment will look like this. Okay, this has a DNA like this, a long DNA is found. And if we draw it in detail, then you will see that at certain points the condensed structures are found. If we draw the diagram of this condensed structure in more detail, it is going to be look like this. And this is the DNA. And this DNA will form some of the RNA and proteins. And there so many DNA copies are found, and these DNA copies remain within this structure. And this structure is known as Balbiani puff. Okay, so such Balbiani puffs are formed. Because of this, we call it as a Balbiani puff. Okay, so now if we draw this chromosome, if we talk about this chromosome are 2 millimeter long have been observed. And in polyteny chromosome, number of chromonemata or fibrils increases up to 2000 or more per chromosome. Okay, so this chromosome have the so many Balbiani puffs or rings, and that form synthesize mRNA, and due to presence of this large Balbiani puffs, so many Balbiani puffs, these are known as giant chromosome. Okay, so this is what about the different types of chromosome in detail. Okay, so if we talk about some MCQs, the part of the cell which is called a cell brain, obviously we know that this is nothing but a nucleus. The nucleus is concerned with the synthesis of, obviously nucleus is having a DNA, so this DNA has the information for the protein, so it is involved in synthesis of proteins through mRNA. Nucleus is concerned with, it is ultimate control of cell activities. Okay, so this is all about the remaining part of the nucleus which we need to discuss, that is being completed. Okay, I hope you understand that. Thank you.